Thank you for joining us today on our Expert Perspectives on Attaining Publishable Western Blot Data webinar. Today we're going to be looking at 3 different perspectives on obtaining robust and replicable Western blots. And we're going to be looking at the perspectives from publishers, reviewers, as well as industry. Dr. Kaoru Sakabe, the Data Integrity Manager from the Journal of Biological Chemistry, will be giving us the publisher's perspective on what can be done to obtain a higher quality Western blot. Dr. Matthew Summers is an associate professor from The Ohio State University, and he'll be giving us a reviewer's perspective on best practices for submitted Western blot data. And I'm the Senior Product Marketing Manager with LI-COR Biosciences, and I'll be discussing what industry players are doing to address the perspectives coming from both publishers as well as reviewers. Throughout the webinar today, Kaoru Sakabe, Matthew Summers, and other panelists will be available to answer any questions that you may have. If you have a question, feel free to type it into the question box. So let's get started with Kaoru. As Kaoru is getting ready, let's do a quick poll. How many of our audience members are familiar with the new Western blotting best practices that have been put forth by various publishers? If you could take a few seconds to give us your feedback, that would be great.
Hello, everyone. First, I'd like to start off by thanking you all for attending this webinar, as well as thanking LI-COR for inviting me to present. My name is Kaoru Sakabe, and I'm the Data Integrity Manager for the American Society for Biochemistry and Molecular Biology. Our society publishes 3 peer-reviewed journals: the Journal of Biological Chemistry, Molecular and Cellular Proteomics, and the Journal of Lipid Research. Today, you're going to hear perspectives from different areas of the research ecosystem on best practices and probably the most ubiquitous technique in biochemistry, Western blotting. Perspective that I'm going to share with you is the publisher perspective. Now, I find Western blotting a lot like the Wild West of the 19th century United States, characterized by Hollywood as an untamed frontier in a period of lawlessness. There doesn't seem to be a set of standards or rules that researchers are taught to apply when designing experiments using blots or when presenting their blot data. And not having a standardized way to perform and present these types of data can cause problems later when others try to reproduce your results. So why is this an important issue for us? Well, from my perspective, or the journal perspective, we see a lot of data at our flagship journal, JBC, that uses gels and blots. In fact, when we analyze the types of data we published between 2008 and 2016, you can see that our bread and butter is gel or blot data. So for us, our main concern is ensuring that our publications are reproducible and will stand the test of time. So here is a brief outline of the topics I'm going to cover. The first topic I'm going to discuss is antibody validation. Now, this is something that should be done for every new antibody you use in the application in which you'll be using them. Remember, there may also be lot-to-lot variations in the antibody, whether you purchase them from a commercial supplier or purify the antibody yourself, which means that each new batch needs to be tested to ensure they're performing as expected. Now, there are a number of reasons why reagent validation is so crucial to perform. First, you may have limited sample, and you don't want to waste it by performing an assay that's doomed to fail. Now, you may save yourself a week by not validating your antibody, but if the antibody ends up not recognizing its target, you may end up wasting precious time and samples, as well as publishing irreproducible research. In fact, that's what happened to this group of authors who ended up retracting a paper when they discovered that many of the key antibodies used in their study were nonspecific. So these are the validation criteria that publishers want their authors to demonstrate. We want authors to demonstrate specificity of the antibody. That is, does the antibody recognize and bind to the target? The antibody must also be selective, meaning in the presence of other proteins, can the antibody still recognize its target? Finally, we want it to be reproducible. The antibody needs to be uniquely identified with the catalog number or a research resource identifier. The assay conditions, such as dilution, blocking conditions, wash buffers, should be described, as these are going to affect staining. And really, if you think about it, this is all information that's useful to everyone, not just publishers. So how do you go about demonstrating specificity and selectivity? Well, in 2016, an international working group for antibody validation proposed 5 pillars by which to validate antibodies. Ideally, you should validate your antibody using a combination of these methods to really demonstrate to yourself that your antibody is specific and selective. Now, there are pros and cons to each one, which in the interest of time I won't get into, but for further information, I would highly recommend reading this article. So with that said, if you run a gel and see a single band by Western blot at the correct molecular weight, it's simply not enough to validate it. You should employ a few other methods that were described in the previous slide to really prove to yourself that your antibody is specific and selective, such as using a knockout cell line. If you're using an antibody that recognizes a post-translational modification, make sure you're using an appropriate control, such as phosphatase treatment for a phosphorylated protein. So the next 3 topics I'm going to go through rather quickly as they are all related to one another. The first one is linear range. So I'm going to guess that the majority of you performed either a Bradford, Lowry, or BCA assay to measure protein concentration of your sample. And if you've done tons of them, I'm sure you've gotten frustrated when your sample falls outside of the linear range of your standards, and you have to rerun your assay because you can't accurately determine how much protein you have in your sample because it's outside the linear range. So that same principle applies if you've overloaded your samples or overexposed your blot. So in this example, I'm showing you 2 different exposures of the same blot. The upper blot has been completely overexposed or is not in the linear range. And if you quantitate the bands, you won't be able to detect any differences. Now, if you take the more appropriately exposed image, you can see that there are now differences in the amount of protein you're detecting. You would get the same type of error if you overload your samples. So being in the linear range is important because it means that the output, or the signal you're detecting on your blot, is proportional to the amount of target in your sample. So being in the linear range segues into the next portion of my presentation, which is about housekeeping protein validation. So in order to demonstrate that blots have been equally loaded, many researchers often probe the membrane with a housekeeping protein such as actin or tubulin. The problem with using an abundant protein is that it often saturates at low concentrations, whereas most researchers are typically loading high concentrations of protein to detect their target. This means that the blot is not in the linear range for the loading control. Another issue is that researchers assume that whatever protein they're using for loading control doesn't change with whatever experimental condition they're using. However, in this study, Eaton and others demonstrate that many of the commonly used loading controls do in fact change with various experimental conditions. Which brings me to my next point about normalization. When you're normalizing signal intensity, you want to normalize it against something equivalent in order to draw conclusions from your experiment. So if you're looking at expression of protein in a sample, the best method for normalization is to use a total protein stain. First, it's linear over a much broader range. And unlike commonly used loading controls, a total protein stain isn't going to vary with experimental conditions. Most importantly, normalization to total protein represents exactly what you're trying to demonstrate. That is, you are showing the expression of your protein of interest against the total protein amount in your sample. Well, how about if you're looking at a post-translational modification on protein, like here? Here, the authors treated cells with 2 different drugs and examined phosphorylated proteins A, B, and C, and normalized it to actin. So the problem with using actin is twofold. First, as we've already seen, the expression of common loading controls like actin may vary with experimental treatment, and if the gel is overloaded, it would be difficult to determine if in fact actin levels vary. Second, remember we want to be able to compare equivalent samples. If we want to show that phosphorylation of protein A increases or decreases with drug treatment, then we really need to see the total levels of protein A. So just to briefly summarize the last 3 sections, make sure you're working in the linear range. You should normalize target protein to total protein using a stain like Kumasi or Ponceau S. If you do decide to use a housekeeping protein, you should demonstrate that its expression does not change with whatever experimental condition you're using. If you're using an antibody that recognizes a post-translational modification, you should normalize that blot to total levels of target protein, ideally on the same membrane. So the next topic I'm going to discuss is replicate analysis. So let's say you're ready to write up your results, and you make your figure by showing a set of representative blots with the summary statistics. What's the best way to show that quantitative data transparently? Well, if you have a small n, it's best to show each individual point by plotting the data using a dot plot. If you have a large number of data points, then I would recommend using a box plot or violin plot. Make sure in the figure legend you also describe the number of independent times the experiment was performed and whether you looked at biological or technical replicates. So the final topic I wanted to cover is about your image data. The JBC, as well as many other publishers, are scrutinizing all the image data in a paper before it's accepted. If images aren't saved properly or if figures aren't assembled properly, this can lead to delays in publication. So your ideal image file is going to have these attributes. Because most publishers will request that you assemble your figures at 300 dpi, your image file should at the very minimum be 300 dpi. If you've used an imaging system to acquire your data, make sure the binning is set one by one. You should save the image as a TIFF file. And finally, make sure you're saving your image either as a grayscale or color. Do not use index color, as this will compromise the quality of your image. So once you acquired your data, make minimal adjustments to it. While I realize there are lots of tools in Photoshop that can beautify your image, much like these apps for your mobile phone, remember the image that you present in your figure needs to be an accurate representation of your experimental results. I would also use software meant for figure assembly, such as Canvas, Photoshop, Illustrator, or CorelDRAW to assemble your figures. PowerPoint is not recommended for manipulating high-resolution graphics. And if you don't have access to the software I just mentioned, don't worry, these programs are open source. So, as you're assembling your figures, remember these key points. If you have to remove irrelevant lanes of a block, remember to declare any gel splices with a line. Also, you can splice together lanes. You can only splice together lanes if they are run on the same gel. Remember to include the positions of your molecular weight markers and show sufficient area around your band of interest. Ideally, you should be able to indicate at least one molecular weight marker above and below your band. Maintain the aspect ratio of your image. Stretching or compressing in just one direction doesn't treat your pixels equally. Finally, don't eliminate the background. Background is a sign of true biological data. So just to summarize, remember to validate your reagents. You should always work in the linear range. If you're looking at protein expression, normalize to total protein rather than a housekeeping protein. If you're analyzing a post-translational modification, remember to normalize to total target protein. If you do use a housekeeping protein for normalization, make sure you demonstrate that expression doesn't change in your system. Plot your data transparently by showing the true variability of your data points. And finally, regarding your images and figures, use appropriate software and remember that your final image needs to be an accurate representation of results. So I know I covered a lot of points rather quickly, so I wanted to close by showing you some additional resources we have available from the JBC. And with that, I'd like to thank you for your attention.
All right, thank you so much, Kaoru. That was a great foundation for today's webinar. Next, we're going to have Matt present on a reviewer's perspective on what is being looked at when Western blots are involved in a research paper. As Matt's getting ready, let's ask another quick poll question. So the poll question is, have you ever been asked by a reviewer to provide more information on Western blot details or about the Western blot data you've submitted? Take a few seconds to give us your insights.
Hello. I'm going to talk about the points that Kaoru has just mentioned for Western blot rigor reproducibility from the reviewer's perspective and give some examples from real-world publications that highlight the importance of some of these points. I'll use the same order of points that she did and begin with antibody validation. So again, it's critical to validate the antibody and report the conditions under which the antibody is used. It's also helpful to include both positive and negative controls to prove to the reviewers that the antibody is doing what it's supposed to be doing. And this is particularly important if the antibody sees multiple bands or if the protein's molecular weight may vary between cell lines or tissues. And just as an example of the importance of conditions and antibody here, we can see using ubiquitin-specific antibodies that some of these antibodies are unable to see free ubiquitin very well, monoubiquitin, whereas they may all be able to see polyubiquitin to varying extents. And to highlight the importance of conditions, this particular antibody, VU101, is actually able to quite robustly see the monoubiquitin or free ubiquitin, under certain conditions. And so it's important to report these conditions, because if someone were to try to repeat your experiment seeing free ubiquitin under the wrong conditions, they obviously wouldn't be able to, and would have some issues likely with the work that you've published. Just further validating that your antibody sees what you think it's going to see, or that it can see what you think it's going to see, is important, and again highlighted by a ubiquitin panel here. So we're looking at the ability of polyubiquitin antibodies, specific antibodies, to see ubiquitin linked through different chains. And you can see that depending on the chain linkage, several of these antibodies have different affinities for the different kinds of ubiquitin. And then importantly, though, despite their all being polyubiquitin binding antibodies, one of them is unable to actually see this one particular M1 linked ubiquitin chains. And just in a second example, this is from a paper that was generating antibodies to GABARAP L1 using 293 cells, which had low expression of GABARAP, to show that their antibody could specifically detect GABARAP when it was put into 293 cells, but then they compared it to a commercial antibody, which, while it was able to detect their protein, also quite efficiently saw the related GABARAP. And so this is something that's of great importance to validate that the antibody is seeing in your cells what you think it's going to see. And obviously, another way to go about that would be an RNA interference or a knockout cell line to show that the signal goes away when the protein is indeed not there. And this is just an example, I think, of a well-controlled experiment. So in the left panels, they're validating that their polyubiquitin antibodies are seeing polyubiquitin and not free ubiquitin, whereas their total ubiquitin antibodies sees all. They validate that they can see a monoubiquitinated protein without seeing it by the polyubiquitin antibody, and then they see that when they stimulate their cells, they're able to identify what looks like polyubiquitinated proteins, which one might think were polyubiquitin chains, but it turns out to be multi-monoubiquitination, as despite the fact that this is a high molecular weight smear, it's not observed by the polyubiquitin antibodies, which they have validated pure proteins, and then showed again under their experimental conditions that a polyubiquitinated protein is efficiently seen by both antibodies. So with respect to linear range validation, I think that reviewers are often still just kind of looking to see if the data in the blot looks to be within the linear range. It's not obviously over exposed or underexposed, allowing us to believe that the data is accurate. But this is starting to change, and reviewers certainly are aligning with publishers who are looking for greater demonstration that we're within the linear range. And so one thing that you might do is just include a demonstration that you have a linear range within your antibody, or at least within the blot. Show us a brief titration just to show that you can detect a range of signals accurately with the antibody. And then, as Kaoru mentioned, housekeeping proteins are still often used. I'd say they're probably still the norm in most of the manuscripts that I look at. But it's important to understand that they can vary with different tissues or different treatments. And in this example, you can see that the housekeeping genes are changing with the stimulus as well as the experimental protein. And if we use a kind of total protein as Kamasi, we can normalize the housekeeping proteins and again see that yes, the stimulus of IGF is changing common housekeeping genes 1.5 to 3-fold. And if you were to use that to normalize as opposed to total protein, you go from having no effect of IGF on EEF2 expression to potentially a 3-fold increase in EEF2 expression relative to a Kamasi stained gel. And so it's important to validate that the protein that you're going to use is actually remaining steady or accommodate for the differences that you're going to be using it for, so that the data is accurate. And in terms of normalization, I'll reiterate a point that Kaoru made about the importance of using the target protein to normalize for a phosphospecific antibody or any PTM antibody for that matter. matter. So in this case, this is an example I pulled. This is not analyses that were performed in the paper. It has nothing to do really with the data that was generated and discussed in the paper, but serves as a nice example to illustrate this point. So looking at activation or phosphorylation of CHEK1 after depletion of a number of ubiquitin-specific proteases, we see that loss of USP7 results in a downregulation of CHEK1 protein. And if one were to normalize CHEK1 protein, or phospho-CHEK1, excuse me, to either CHEK1 itself or the loading control, both of them will give you the result that there's a dramatic loss of CHEK1 phosphorylation in the absence of USP7. However, if we normalize those to the CHEK1 phospho-CHEK ratio rather than to SI control, we see that that difference is actually fourfold greater if we use the loading control rather than CHEK1 itself as a normalizer. And then it's also important to be clear about the nature of presented controls. So ideally, the controls will be from the same gel in which the experimental blot was made. But, and I think it's important to note, and we'll hear more about this from Jeff, that recent advances in imaging and multi-channel imaging and total protein detection are making this much easier, as shown in these examples here, to provide this information for our reviewers or our public that we're presenting our data to. And if, for example, the protein are run on different gels, so the loading controls are run on different gels for various reasons that might be necessary, it's important to point out to the reviewers that this is possible, because it can cause some uncertainty about the nature of controls and what the reviewers are looking about. And this example was pulled from a sort of watchdog site, pubpeer.com, in which it appears that the phosphoprotein bands were run on different gels than the total protein and loading controls. There could be many reasons for this, but it caused concern to the readers and potentially to a reviewer, where you can see that there are differences between them. There are different patterns of spacing between the lanes. There's different patterns to certain smiles in bands between the different gels, and so it can cause some concern. So it's just important to point out that that was the case if indeed you're running protein panels from different gels. And then in terms of replicate analysis, I think just one thing I would point out, and that is to be sure that the blot that's being shown as representative of the quantified data that you've quantified your replicates is the most representative of those. And in this example here, we can see that under different conditions, we have different levels of proteins A and B. And if you were to measure these and you plot the replicates, you see that conditions Y and Z, by and large, have a very similar impact on protein A. And this is probably actually more similar if we were to look at the individual data points, because you can see there's probably two that were a little bit of an outlier in these that changed what was otherwise very similar results between the two. However, that's fine, and the transparency is good, but it's still a little misleading to show a blot that doesn't represent the actual take-home from the data very well. And then finally, a few brief words about image data. Again, I think it's important to reiterate that we want to work within the linear range. You don't want to present blots that are overexposed or oversaturated, because it calls into question the ability to quantify and whether something is being hidden. And similarly, it's important to avoid too much processing. While some Western blots may be very beautiful, they aren't perfect, and I think we as reviewers expect that there are going to be some imperfections and blots and bands, and if something has to be highly processed in order to hide those things, if it looks doctored, one wonders to what extent it's doctored and why it's doctored. And if the reviewers don't flag it, I think that most of the time the publishers will. And then along those lines as well, it's important to, if you do need to remove lanes, it's best to try to present your gels without having to do that. But if you do need to remove intervening lanes or between the data that you want to present, that's often quite readily observed by the reviewer or the reader, and it's important to point out that you've done that so that one isn't left to wonder what it was that you were hiding or whether these two pieces actually came from a similar gel at all. And then lastly, it's easier said than done, but it's good to strive for well-resolved and individualized bands to increase the confidence of the readers in any quantification that is being done based on the Western blot data. That's all. Thank you for your attention, and let Jeff move on with the next phase of the webinar.
All right. Thank you so much, Matt. Now that we have a solid understanding of the best practices from publishers, as well as insights into what reviewers may be looking for, I'd like to take the next 10 minutes to take a look at what industrial players are doing to address those different perspectives. Now, before we do that, let's do one final quick poll question. And the question is, what software packages do you traditionally use as you're preparing your Western blot data for publication? You can check all the answers that apply. So given these best practices and knowledge of what publishers and reviewers are looking at, what can be done from an industrial perspective to make your Western blots more reliable? There are several areas I will briefly look at today with regards to what can be done to generate higher quality Western blots. We'll be looking at your detection method, your reagents, your software, your protocols, as well as your training. So let's begin with the detection method. One of the first steps that you can take to generate more robust Western blots is to move from film to digital imaging. This is going to allow you to move from a single color to multiple colors, which allows you to do proper normalization techniques for quantification. Secondly, you can get better data by simply moving from chemiluminescence to fluorescence. Now, chemiluminescence, as you know, is a dynamic enzymatic reaction, and that can add some challenges to getting the same signal response from all of your experiments, or let alone if there's another lab that's trying to replicate your data. A third area to look at in digital imaging is the dynamic range of your digital imager. Now, Odyssey imagers from LI-COR provide over 6 log of dynamic range, and this allows you to capture the full range of signal, the weakest bands to the strongest bands, all in a single acquisition without ever resulting in saturation. Now, this is important because in this way you can accurately assess the linearity of both your loading control as well as your target. This is what we call the combined linear range. And so when you are accurately assessing the combined linear range, you can empirically calculate how much protein you should be loading onto your gel, so later you can accurately quantify the signal. So those are several areas with digital imaging that can help you get more accurate data. But that's not the only steps we need to look at. Reagents can also be a big source of variability. Now, when you're using reagents, as Kaoru and Matt have already discussed, you need to make sure that you're validating those reagents. Use reputable suppliers that provide you with all the information that's necessary for your publication. And when you get those reagents, also, don't take anything for granted. Make sure that you optimize those reagents for your system. The next area we're going to take a look at is the software. And this is one of the most important areas to consider because oftentimes it's overlooked or not considered as a variable. Software can often lead to large variations in the results if it's not clear on how the data should be analyzed, or if there's too many ways to analyze the data, or if the software is actually altering the data. Now, Imperia Studio software is the software LI-COR has designed in light of publisher and reviewer best practices. The areas that Kaoru and Matt have talked about have been built in and integrated into this software to ensure that your data is robust and replicable. And in addition, it's in alignment with what they're going to be looking for. For example, Imperia Studio keeps track of all your antibodies as well as the critical information about those antibodies. It also contains any validation experiments that you've done, ensuring those are ready for submission as supplemental data. Now, many labs will not empirically determine how much protein they should actually be loading onto the gel. And this can be challenging to a certain degree because you have to determine the linearity for both your target as well as your loading control, what we call a combined linear range. Now, Imperia Studio makes that really quick and really easy because it takes those two experiments and it tells you how much you should load, and it also conveys if you don't have a combined linear range. For example, on the left, we see an example where the loading control and the target do not have a combined linear range, as seen that there's no combined area where green reflects that linearity. On the right, on the other hand, we do find a combined linear range for both your target and loading control, and it tells us that we should load 5.5 micrograms. Now, this is going to be very useful supplementary data for publishers as well as reviewers because it'll tell them that you did all the calculations and they can trust the calculations of fold change and quantification differences. Now, for those of you who are using a housekeeping protein as a loading control, Imperia Studio also quickly walks you through that housekeeping protein validation. That way you know and can demonstrate that the housekeeping protein you're using is valid and will not change based upon experimental conditions. Likewise, Imperia is designed to quickly walk you through the normalization using any of the three methods, whether it's a housekeeping protein, a total protein stain, or if you're using post-translational modifications such as pan-phospho. Now, Imperia Studio also incorporates the necessary statistics to analyze your replicates so you can accurately determine the CV as well as fold change. There's no longer a reason to export into other programs like Excel or GraphPad. And finally, the graphs and charts that are created by Imperia Studio are generated and put into a report with your data. And all of this is in alignment with what publishers have suggested for submitting. Now, one of the other areas where quality can be compromised besides software is protocols. And frankly, there's probably as many different protocols as there are researchers. And unfortunately, many of those may not even be in alignment with what we currently know about obtaining that higher quality data. Now, the protocols that LI-COR has put together have been written in light of today's best practices from journals such as JBC. And finally, training can be a very important issue in obtaining high-quality data. Now, oftentimes in a lab, there's not a consistent method of training for the lab members, and many times if there is a lab training in process, it may not be in alignment with today's best practices. For this reason, LI-COR has put together Lambda U. Lambda U is a 24-hour-a-day, 7-day-a-week training portal that's accessible by you. All of the courses are based on the best practices that we've been talking about today. And it includes not only the theory behind what you're doing, but also practical in-lab demonstrations. So in summary, Western blot best practices are critical for every lab, including yours. And frankly, it's never been easier to ensure that your lab is incorporating these best practices. So as you're assessing how your lab measures up, take into consideration your detection method, the reagents that you're using, the software you're working with, the protocols your lab is using, and finally, determine how you're training your current and your future lab members. Now, if you'd like more information on how LI-COR can help you out in these various areas, I'd encourage you to reach out to us. Thanks so much. All right. Well, thank you, Kaoru. Thank you, Matt, for presenting today at today's webinar. And so at the next step, what we'll do is take a few moments to see if there's any questions out there that we could answer. All right, so the first question we have here is regarding Lambda U and whether or not Lambda U is free. And the answer to that is Lambda U is both free and charged. It depends on what you're looking at. So there are some courses in there that are free to the world. Some of the courses are for charge, and then LI-COR customers that actually have our equipment, when they order that equipment, they do get some free passes to get to a variety of the courses as well. So it depends upon where you're at in the subscription level. The next question that we have is whether, I believe this is with regards to Imperia Studio, and whether or not that can be used with other imagers. Imperia Studio has been designed to work around the Odyssey family of imaging systems, and so it would not be able to be used with other instruments at this point. All right, so at this time I think we'll begin to wrap up, but we'll still be online answering any incoming questions, so if you think of a last-minute question, feel free to go ahead and type that in. We do thank you for joining Matt, Kaoru, and myself today for the webinar, and if you have any other questions, you can always reach out to LI-COR. Thanks a lot.